We really wanted to find a way to prevent the development of end-stage kidney disease requiring dialysis and transplantation. To do that, we needed a source of the cells that produce insulin.
Bob Evans Farms became engaged with the Rogosin Institute, actually through a phone call that I returned to Dr. Rubin. After listening to Dr. Rubin and understanding how we could become involved in the research, and also meeting Dr. Rubin and Dr. Jean in person, it became a very easy decision for us to make to get involved in supporting the Institute in any way we could. The hogs that we harvest for manufacturing our sausage products are the ideal age for isolating islets from the pancreas glands.
We, with Bob Evans' help, built a building on property that belonged to them, and that launched the real effort both in diabetes and cancer.
What started out as a small 400 square foot laboratory over in our sausage plant went through several generations and ended up as a 10,000 square foot freestanding laboratory here on Bob Evans' property, as well as a much expanded laboratory over in our plant. Our hope is that we'll have a Rogosin laboratory at all of our plants.
The problem with diabetes, if you want to treat it successfully, is that yes, you have to replace insulin, but you have to replace the other substances that are made by the islets, and those are about 120 in number as far as we know. But you can't just take islets from hogs and put them into a human being, because the human being's immune system will destroy those islets before they have a chance to do anything. So what we do is we encapsulate the islets in a shell made out of a sugar from seaweed, and that capsule protects the islets so that they are not destroyed by the host or the patient's immune system. We expect to be able to put the beads into human beings with severe diabetes. We began to think about what other cells we might put in the beads to treat diseases other than diabetes. And we found, in fact, that cancer cells did very well in the beads, and that they produce factors that inhibited the growth of tumors and cancer cells outside the beads.
Up to now, we've been able to make these cancer macrobeads by hand, and that's been sufficient to meet our early stage clinical trials. The automation equipment that we have here in Xenia now, or the robots that have been specially developed for our process, will allow us to greatly increase the production of these cancer macrobeads and thereby treat many more patients. A lot of cancer research today involves cancer cell lines, which are normally grown in a culture dish in a two-dimensional environment. So the macrobead offers the advantage of a three-dimensional environment, and I think more naturally replicates a normal in vivo environment. And so this has allowed us to understand some of the basic properties of cancer. For example, the cancer stem cell.
And what we found in treating patients with severe or advanced forms of cancer, that we could control the growth of the cancers and at the same time, very importantly, greatly improve the quality of their lives.
We work very closely with the FDA, and I think we have a terrific relationship with them. They've been very helpful. I think they are as interested in seeing this project succeed as we are. One question that I ask potential new staff members out here in Xenia is, what is it that they're the most passionate about? If our passions align, then I know that I have found the right person.
What we're working with is what I believe is the future of the treatment of disease, which is not with specific drugs designed to target an enzyme or a receptor or whatever, but we're talking about treating diseases by delivering cell systems that can restore body function to normal. We are repairing the body, not just treating symptoms and specific problems.
Many years ago, my wife and I had a child who spent 18 months at Children's Hospital in Columbus. And it's those memories that we have from that very difficult time that makes me want to see this research succeed so that someday, instead of doctors saying we have no cure or there's nothing we can do, that they can say, yes, we can treat that disease or we can treat that illness.
I know firsthand the difficulties that both patients and their families go through with either diabetes or cancer. The potential that either the cancer macrobead or the islet macrobead has to offer is there. And I think that everyone that works here sees it the same. And that is our own personal responsibility to see it through.